Hello students, today we will discuss about the tissue systems. Okay, so what is tissue system? So we discussed already in the previous video about different types of tissues. What is tissue? Group of cells, okay, which may be similar or dissimilar but perform similar function is a tissue. Now, in the different organs of the plants, like root, stem, leaf, flower, fruit, you will find all these different types of tissues together. And all such group of tissues, wherever they are present and working for one purpose, will form a tissue system. Okay, and as we are talking about the plants, in plants we find three main types of tissue systems. They are epidermal tissue system, ground tissue system, and vascular tissue system. Okay, this I am giving you one flow chart here. The epidermal tissue system. Here we will discuss about the epidermis, the protective layer present on every organ of the plants, epidermis, and sometimes you will find some extra structures, and these extra structures are called epidermal appendages, and these epidermal appendages, when they are associated with the root, we call them as what root hairs, and the hairs that are associated with the shoot system, maybe with the stem or maybe with the leaf. Okay, then we call them as trichomes, and sometimes you will find some spine-like things or thorn-like structures, but they are neither spines nor thorns, and we call them as what emergences. Okay, and the best example is rose plant, and in the rose plant you will find many pointed structures, and the people mistake them as what some people call them as thorns and some people call them as spines. What I am telling you is that they are neither thorns nor spines. Then what are they? They are prickles, and they fall under this category. We call them as what emergences. Okay, we will discuss them in detail later. And now let us see the ground tissue system. The ground tissue system is present between the epidermal tissue system and the vascular tissue system. So the ground tissue system can be classified into two types: extracellular ground tissue system, intracellular ground tissue system. And this extracellular ground tissue system includes hypodermis, general cortex, endodermis. And intracellular ground tissue system, that is the central vascular cylinder, is called the stele, and that is surrounded by a tissue called pericycle. So pericycle, and in the center of the stele you will find one tissue, and that is called pith or medulla, and you will find some elongated cells in between the vascular bundles, which are called medullary rays. So that is the ground tissue system. And when we come to the vascular tissue system, the vascular tissues are xylem and phloem, and they are arranged in the form of bundles in the primary plant body. Okay, whereas they are arranged in the form of rings in the secondary plant body. So these vascular bundles in the primary plant body, in roots, they are arranged separately on separate radii. So we call them as radial vascular bundles. When you find the xylem and phloem together, okay, then we call them as conjoint vascular bundles on the same radius, xylem and phloem. And based on the number of xylem elements and or number of xylem strands and number of phloem strands, They are further classified into two types: collateral, bicollateral. And in collateral, you will find one xylem strand and one phloem strand. And in bicollateral, you will find one xylem strand and two phloem strands. That's why it is called bicollateral. And we have the third type, that is what concentric vascular bundles. And these concentric vascular bundles are of two types: amphivasal or leptocentric, amphicribal or hadrocentric. That means here the vascular bundles are arranged in the form of a ring. Means what? One will be surrounded by other. And you can see here leptocentric. Leptome is the other name of phloem. Centric means what? Center. So when phloem is present in the center, surrounded by the xylem, that is called amphivasal or leptocentric. Hadrome is the other name of xylem. So you just remember these two terms. Hadrocentric means what? Hadrome, that is xylem is in the center, means it is surrounded by the phloem. That it is called hadrocentric. So these are concentric vascular bundles. Okay. First, you just memorize this flow chart, and we will discuss first about the epidermal tissue system. So now let us discuss the epidermal tissue system in detail. So the epidermal tissue system is composed of epidermis and appendages. Epidermal appendages. First, let us discuss about the epidermis. Here, I would like to discuss the epidermis separately. Of course, we know that the epidermis is a protective layer present on every organ of the plant. Okay, so epidermis is present on root, epidermis is present on the stem, epidermis is present on the leaf. So generally we can call the epidermis of the root as root epidermis, stem epidermis, and leaf epidermis. So they have some variations. Of course, many similarities are there, but at the same time, you will find some dissimilarities also. So, this epidermis is made up of what cells actually? The epidermis is made up of parenchyma cells. So, this is a living tissue or a dead tissue? Living tissue. So, epidermis is a living system. And these parenchyma cells, in all of them, whether it is root epidermis or the stem epidermis or the leaf epidermis, you can see here, they are arranged in a layer. So generally, they are uniseriate. Of course, multiseriate you will find in some xerophytes. But here, you can see that the cells are arranged in one layer. And you can see that they are arranged compactly. Compactly means they are arranged very closely. So whenever they are arranged closely, what will be absent? The spaces are absent. So in the epidermis, intracellular spaces are absent. Okay. And let us see, in all of them, these epidermal cells are parenchymal cells. They are arranged in one line, and they are arranged compactly without intercellular spaces. This is applicable for root epidermis, stem epidermis, as well as leaf epidermis. But in leaf epidermis, one exception will be there. Means what? In the leaf epidermis, here and there, you will find some gaps. These gaps are called stomata. And these stomata may be present even in the stem, but they are very few in number. So the stomata are mainly okay concentrated in the leaf epidermis. That's why I am showing the stomata only on the leaf epidermis. Okay, and here I just say some of these root epidermal cells. I am talking about this one. You just see here. This root epidermal cell elongated, and this elongated structure is called root hair. Root hair. So you will find such elongated epidermal cells only in the roots, and which are called root hairs, and they will increase the area of absorption. Increase the area of absorption means what? The epidermis can absorb water and minerals, but to increase that area, because you remember that even though the roots may be very long, but the entire length of the root will not absorb the water. Only at one particular region, the root will absorb water and minerals. That region is called what? Root hair region or maturation region. Okay, and of course, this epidermis with root hairs is called epiblema or rhizodermis. Epidermis with root hairs is called rhizodermis. Now, I hope you are understanding this point. So, suppose if anyone asks you that epiblema is associated with, then what you have to say? Root. What is epiblema? If anyone asks you, the epidermis with root hairs is called epiblema. Okay, and what is the function of epiblema? Okay, it due to presence of root hairs, it will increase the area of absorption. So that's what the thing. But some means what we have to compare the root epidermis with the stem and the leaf epidermis when it comes to the comparison. So what we have to say? So here you can see the stomata, and I told you that even stem epidermis may have stomata, few stomata, and here you will find one line also. And what is this line actually? So this represents the waxy coat or waxy layer called cuticle made up of cutin substance. And do we find that cuticle here? No. So what are absent in the root epidermis? Cuticle is absent. Okay. Stomata are absent. Because a root is not transpiring organ, it is absorbing organ. And if cuticle is present, so it will okay, it will not allow the entry of water inside. That's why they are not provided with sorry, cuticle. Okay, when we come to this one, okay, here I drew two diagrams. One is let us say this is dicot stem epidermis, and this is monocot stem epidermis. So in both of them, the cells are parenchyma cells. So they are arranged, they are rectangular cells, and they are arranged compactly without intercellular spaces. Okay, but they are provided with a waxy coat called cuticle that minimizes the rate of transpiration. But the dicot stem epidermis is provided with one more structure, that is what the epidermal outgrowths. Epidermal outgrowths, which are called trichomes. So just remember, trichomes are present in dicot stems and trichomes are absent in monocot stems. Okay, when we come to this leaf, okay, the leaf epidermis is also made up of parenchyma, and the cells are rectangular cells, and they are arranged compactly without intercellular spaces. But some cells are okay modified, and what are those cells of the epidermis that are modified guard cells. I hope you're understanding. These are guard cells, and the adjacent cells are called what? Subsidiary cells. They are different from the rest of the epidermal cells. Okay, and you can see here. So this is the dicot epidermis, dicot leaf epidermis, and this is monocot leaf epidermis. Okay, and here you can see that what is the extra structure you are finding here? Trichomes. So trichomes are present in associated with dicot leaves, and trichomes are absent in monocot leaves. But both are provided with the cuticle. So cuticle is present in both. But one more point I hope you are noticing here in the from the diagram. What is that? So I put some dots in the guard cells. What are these dots represents? Chloroplasts. So remember, epidermal cells are actually devoid of chloroplasts. Means what? Chloroplasts are absent in the epidermal cells. You can see here. Okay, nowhere I put the dots, but only in the guard cells of the epidermis here, I put dots. So the dots represent what? Chloroplasts. They are present only in the guard cells. The epidermal cells with okay chloroplasts. If anyone asks you like that, the answer should be what? Guard cells. And okay, when we come to this monocot epidermis, you will find sometimes, as in the case of grasses, which are monocots, you will find some dead cells, dead epidermal cells, which are hygroscopic in nature, and these cells are called bulliform cells. But of course, these bulliform cells are present in the upper epidermis or adaxial epidermis of monocot leaves. Adaxial epidermis of monocot leaves will have these bulliform cells, monocot leaf. And one more thing. So they have bulliform cells are present in monocot leaf epidermis. Bulliform leaves are absent in dicot leaf epidermis. Trichomes are present in dicot leaf epidermis. Trichomes are absent in monocot leaf epidermis. And now let us see the details of that stomata. So this is stomata, and this is in dicots, and this is in monocots. So here also I had to put some dots. So these are guard cells, and this pore is called stoma. Okay, and these are accessory cells or subsidiary cells. Okay, so actually these two are the epidermal cells. So which are bean shaped or kidney shaped in dicots. So the guard cells are bean shaped or kidney shaped in dicots. Bean or kidney shaped. And their arrangement or their orientation is also special. That means they exhibit special method of arrangement. They are not arranged like this. So you can see that they are arranged like this. And as these two cells are arranged like that, so a space is formed between them. So don't forget that what we said. So generally the epidermal cells are arranged compactly and intercellular spaces are absent. But in this area you find the intercellular space, and that space present between the two guard cells is called what? Stoma or stomatal aperture. Stomatal aperture. And you can see that these guard cells are provided with what? The chloroplast. Okay, so only guard cells will have the chloroplast, and the surrounding cells, okay, are the cells which are in the vicinity of the guard cells. Means what? Close to the guard cells are called accessory or subsidiary cells. But of course, remember that this stoma plus guard cells plus these accessory subsidiary cells, all of them work together. Okay, they regulate the transpiration. That's why this is called stomatal apparatus. Stomatal apparatus. So now tell me what is meant by stomatal apparatus. Stomatal apparatus includes the stoma or stomatal aperture plus the two guard cells and the accessory cells. And when we come to this monocot stomata, in monocots, the guard cells are dumbbell shaped. These are guard cells, GC, and they are dumbbell shaped. Dumbbell shaped guard cells. And you will find not many subsidiary cells. So here you will find only two subsidiary cells. And I hope you might have noticed one more thing here. So that is what the inner wall of these guard cells is thickened. The outer wall is thin. Because of that, okay, so you'll find whenever the guard cells become turgid, okay, this stoma or aperture will become, okay, so it becomes more concave. And a result of which it widens, and we call that widening of the stomatal aperture as what? Okay, opening of the stomata. And whenever the cells become flaccid, so then this concave shaped one will become what? Okay, less concave. So then it becomes narrow. So remember the point that it never closes completely, partial closure. Okay, and that is because these stomata not only help in transpiration but also help in gaseous exchange. So that is what about these stomata, epidermis.